So I'm going to bring it back to people and mice together this time to break the pattern a little bit. I'm going to tell you about why mice are not little people, for example. Okay, so let's imagine for a second that you're diagnosed with an incurable disease, right? Which is unfortunately not unlikely. But there's a good news. There is a candidate therapeutic, and there's an ongoing clinical trial that just started. So you get excited, then you look it up and learn that it's about maybe 5–10% chance that it's actually going to be successful, and that's going to take 9 years. So I could tell you that there might be a different way, and you could get your answer, a diagnostic or therapeutic candidate, within months if you are willing to sacrifice about this much of the tissue, not to scale. A grain of rice, several cubic millimeters, that will be extracted from your body and will actually give you the answer for the drug that works for you. And that probably sounds like a pretty crazy statement because they are used to drug discovery being this really long process, right? We have to understand the disease first, find some target, put it into mice, and only eventually put it into a human. And in the process, we're going to spend that 9 years, fail vast majority of the time, and then spend $1 billion when include all the failures, and you need to understand the disease before you even can start that process. But there could be a different way. If you could just go to the last step and resolve it right there, you could save a ton of money, time, and cure a lot of people. But the key is how to do it safely. So how do we make sure that, for example, for dosing the drug to a very small piece of tissue, it doesn't diffuse around? You can sacrifice the safety of a cubic millimeter, but not the whole human. And if you were able to test large amounts of drugs in that cubic millimeter of the tissue, you could throw the kitchen sink at the problem and find the answers, even if you don't understand why it works. So how do you do that? Of course, there's more than one way to do that. I'll give you just one example for one kind of disease, but could be generalized to other indications. So imagine that you have a vitiligo, which is a common skin disease. Then you inject a library of gene therapy vectors, the same ones that are already FDA approved. Then what you do is use a low dose, and what happens is that the vectors enter different cells, and each of them carries a different drug. The cells are becoming their own drug factories. Then you wait a couple of weeks, and you basically identify which cell turned from the white color to the brown one, and that is a phenotypic cure for vitiligo. So you take every single brown cell, and you see what kind of vector is inside, because that is determining what was successful. You already have a drug that has worked in that skin in the person. So by simply looking for successful cells, you have your answer even if you don't have any idea how it works. So of course that could be generalized to different organs and diseases, and you can use it even to de-risk some aspects of toxicity. I ran a bit of a study trying to understand what could be addressed with this. And here are some 4 examples. This is one of the dozens that we identified, where, for example, vitiligo, fatty liver, ALS, and pulmonary fibrosis are all fair game because they're on a single cell level. So as long as the disease can be read out on the single cell level, it's a fair game. So among dozens of indications, even just these 4 are going to have $22 billion addressable annual market. Additionally, because you can skip the mechanistic discovery and go straight to the drugs, you could disrupt the $200 billion market of the preclinical discovery, which is monopolized by WuXi with $65 billion, which is going up. Of course, we're aware that there is FDA regulations and there is the need for a paradigm shift, and there is not even a regulation for this because it's very new. But eventually what will happen, I think, is that once you prove that it's safe in the animal, that 1 millimeter of tissue does not affect the rest of the system, that you can find drugs based on the randomized libraries, we'll probably realize that dosing the entire human with a high dose of an untested drug is actually not the best way to go about it. And yet we accept that as a status quo. Perhaps it's better to deploy it in a small piece of tissue and then make sure that it doesn't affect the rest, right? All right, so do we need to make that happen? Well, we have to localize the drug action, and I will show in a second there are ways to do that. We need to learn how to read out large numbers of drugs from that piece of tissue, and we need to test it in various indications and validate it to make sure that it's safe and effective. And there will be some cool benefits that are not obvious, such as that if you get this kind of data, if you find your drug, you can actually see how it works and learn the mechanism of disease. Or you can get real human disease AI training data and actually empower the whole AI revolution so it doesn't use just data that was identified from mice. And there's a lot of supporting technology. Some of this even came from our lab. So I'll give you an example of localizing drug delivery. This is a ball of gene expression from an FDA-approved vector that is contained within 1/4 millimeter of the tissue at 3 weeks' time. What you are seeing on this next slide, which is slightly complicated, is that each of the dots that you're observing here is a different drug design that we tested in a mouse. So this is 4200 that we tested in one animal, But our lab actually made 1.3 billion of these, and it cost about $4,000 because it's actually very easy to do with biologics. And then the last case, this is a cool technology. We have a patent application that's pending on this, which shows that you can tether peptide drug to the outside of the cell. So that makes it easier for you to actually test for drugs that are more deployable because you don't need to put them inside the cell. They just can act on the outside. And what we are showing here is that you can actually bring a cell from a fatty liver cell back to the point where it has less fat and it's comparable to actually a cell that never has been dosed with the fatty acids. And of course, that's a big indication. So what do we need to make that happen? Well, we need to reduce it to practice. We need to develop technology, make sure it's safe, make sure that you can screen lots of drugs, and know how many can we do maximum, how to make sure localize drug action. Then we need to put that into some kind of models to evaluate how well does it actually work. And eventually, after some 5 years or so, we're hoping that we can translate it by making partnerships with pharma companies. Either as a platform that allows us to screen better, or even for specific indications for the most burning diseases that are also technically the easiest. So what do we need to do? If you are tired of seeing those headlines where you see that something has been cured only to find out it's been in mice, and it's been 10 years and never transferred to human, or that you know that there are so many rare diseases for which there's simply no economic case to resolve it because they are just too expensive, then that could be a good idea to consider starting ASAP and just reframing how we do clinical trials, making them safer, and at the same time making them mechanism agnostic and much faster. So I spent about 10 years thinking about it. This is basically, I just got over the tenure at Rice, which means that I can now take risks and I can try to do things with a longer time horizon. So I'm sure you have many technical questions. There's way more ways to do it than just one that I have shown you. So just please talk to me and I'll explain all the technicalities because, condensing 10 years of thinking to 7 minutes is very difficult. So thank you.